So it's my pleasure to introduce to you the Ma Lab, and Huang is going to, I think, introduce the topic areas, and then looks like there's 3 other speakers. So this is pay close attention to everything you're saying. It's fine. Okay, go down.
Hello, testing. Can you try that again?
Testing.
Yeah, it is showing now. Perfect.
Okay.
Go ahead.
Okay, great. Well, hello everyone. Thanks for coming. Yeah. So this is my group, and I guess hello to everyone on Zoom as well. Let me just get used to the controls here. Okay, so this is the title slide. Okay, so we're here to just talk a little bit about what we do and give you a few examples of our work. Okay, so we're a bunch of engineers. We're quite different in some ways in background from everyone. And what we do is we develop new technologies for biological research and for medicine. Okay, and so we look at what kind of developments are happening on the sort of the tech side and try to bring some of those things, bring a lot of those capabilities to biology and medicine. And the particular flavor, particular style that we work at is really we look at single cells as individual functional units. We'll try to look at how do we measure the function of single cells, and then to get some idea of how do single cells work in a particular context, or how do they make decisions, and how that is how that works in various scenarios. Okay, and so the areas that we draw on in terms of basic capabilities from engineering are some of the ones here. So microfabrication. So this derives from a lot of techniques from semiconductor manufacturing, where we can make small scale structures sort of at the length scale of individual cells. Okay, and then there's microfluidics. This is where there's been all this development in the last 20 years on trying to develop ways to control very precisely the flow of very small amounts of liquids. The other thing is there's all this great work that's been happening in microscopy and data analytics. So there's a lot of information that you can get from microscopy, and now even more with machine learning, you can extract that information. So by combining all of these things together, we hope to bring new capabilities for you guys all to do wonderful research on. So today, what we'll do is we'll just present 3 vignettes of the different projects in my group. Okay, so we'll start with Pan, who's going to talk about neutrophil extracellular traps, and then we'll go with Erik to talk about red blood cell deformability, and then finally Jayden will talk about analyzing insulin secretion from beta cells at a single cell level. So, I guess the plan is to take questions at the very end, right? Yeah. Okay.
Okay, so hi everyone. Today I'm going to talk about neutrophils. So the neutrophils are the most abundant white blood cells. They are the first responders of our immune systems, and their activities have been linked to lots of conditions like cancers, infections, and viral inflammatory diseases. And they can perform their functions in lots of different ways, like phagocytosis, releasing antimicrobial cytokines. But today we are focusing on mitosis. After being activated by things like bacteria or fungi, neutrophils could release neutrophil extracellular traps, and we call them nets. They are mostly composed of DNA and histons. They could efficiently trap and kill bacteria and other pathogens. The process of this net releasing is called netosis. During inflammatory responses, neutrophils could form swarm-like patterns in infected tissue, but it is still unclear how they cooperate to eliminate these pathogens. And here's our proposed model. So in the first step here is netosis initiation. It has been proved in experiments in vivo that pathogens at site of infection could trigger netosis of the early arriving neutrophils. Then how about the late arriving neutrophils? These neutrophils cannot get direct contact with pathogens like the early arrived neutrophils. So how they function? So we think probably the early arriving neutrophils, the neutrophils that have been activated by the pathogens, could release signals to nearby neutrophils. So the netosis could propagate from the center of site of damage to form a large nest area, which could efficiently trap and kill pathogens. And to verify that, we need to prove that whether there is a self-induced netosis, so whether stimulating neutrophils could directly trigger netosis. If it could, then we want to identify the self-produced signals which are responsible for this process. And then we want to find out the mechanism of netosis in the swarms of neutrophils at site of infection. Since the neutrophils could migrate around, it would be hard to track their responses over time. So here we use nanowells to isolate them inside the nanowell unit. So we use the PAC-DA 250 hydrogels and a UV laser patterning system for the fabrication of nanowells. So we use the UV light to photopolymerize the prepolymer on glass light. Then we use ethanol to wash out and cure the prepolymer. That's how we get a nice micro well pattern on a glass light. And the final step is just to assemble a well plate frame with the glass light. And in this image here, we could see that nanowell unit size is around 80 times 80 micrometers, and wall height range from 26 to 35 micrometers. And Jayden will give you more detailed information about nanowell fabrication. So for cells that have been treated with netosis inducer, there are around 6 stages of them. Let me bring my laser pointer. So at beginning, those are healthy cells for sure. And for cells that have been stained with hoax, they look super bright. Then the cells exhibit DNA decondensation. At that time, they still have intact membrane, so the fluorescent intensity will still be very high. But after the breakdown of cell membrane, the DNA goes outside of the cell, the fluorescent intensity will drop dramatically. We could also see other things like DNA fragments and some dead cells. And for dead cells, they are quite different from the cells releasing nets because they're super bright here. And for our study, we first start with the DHL60 cell, which is a neutrophil-like cell. And it has been extensively used in the study of human neutrophils. So there is an image from the control group. The cells have been stained with the hoax. They look super bright and rounded in very good shape. But for the cells treated with netosis inducer, we could see some cloudy-like structures, the web-like structures. Those are nets secreted by activated cells. So here we could also see some DNA fragments released from activated cells. Then we needed, for the netosis quantification, we use the automatic cell counting based on their fluorescent intensity, the stained area, and their circularity. So we try to identify the intact cells from activated cells based on, since they have higher fluorescent intensity, relatively small stained area, and good circularity. So here is an example. For image on the left, this is an image from a control group. So almost all cells have been highlighted in red, and they will be treated as intact cells. But for the image on your right, this is an image from a treated group. So we used L-lysine calcium salt as the netosis inducer. We only have some intact cells, and for cells that cannot be highlighted in red will be treated as intact cells, will be treated as activated cells. And to calculate the percentage of cells undergoing netosis, we basically just need to take 2 images. We will take very first image immediately after adding the netosis inducer, since the cells cannot respond that fast. So all cells will be highlighted in red. So that's how we get the total number of cells in our nanowell unit. After around 4-hour incubation, we will take the second image at exactly the same nanowell unit, and then we will count the number of intact cells again. The difference between those 2 images will be the number of cells releasing nets. And with the information from the first image, the total number of cells, then we will have the percentage of cells undergoing netosis here. Here are some results for cells treated with L-lysine at different cell densities. The x-axis here shows the density of cells per nanowell unit, And the y-axis here shows the percentage of cells undergoing netosis. So for the control group, the blue squares here, the cells stayed at a very pretty low level. But for cells from treated groups, we could say that higher density of cells, the higher percentage of cells undergoing netosis. And it also showed a strong correlation between the NES level and the cell density here. So this result suggests that there might be an enhanced cell-cell communication at high cell densities, which amplify this L-lysine-induced netosis. So then we want to see whether the stimulated cells could directly trigger netosis. So we cultured the stimulated cells with naive cells inside null unit. So for the stimulated cells, that's our cell batch 1. We first treated them with L-lysine for at least 25 minutes. Then we removed L-lysine before adding the naive cells. That's our cell batch 2 here. We only stained cell batch 2 with host, because we only care about how many cells in cell batch 2 will go through netosis. And here are some results from DHL60 cells and human neutrophils. The x-axis here shows the cell densities of treated cells, the number of treated cells per nanowatt unit, the stimulated cells here. And the y-axis here shows the net percentage of nets of untreated cells. That's our cell batch 2. So we could say that the higher density of stimulated cells inside nanowatt unit, so more cells in more untreated cells released nets here. So we think that the stimulated cells did release some sort of signals to trigger netosis of untreated cells. And to further study these self-produced signals, we investigate the impact of nets released by stimulated cells. Since the main component of net is DNA, so we use the DNAs, try to digest the nets. And for the group treated with DNAs here, it showed reduced net level of untreated cells. And we also found that there is a great similarity between mitochondrial DNA and bacterial DNA. And both of them could trigger netosis directly through the toll-like receptor 9 signaling pathways. So here we used a TLR9 antagonist here, ODN-A151. And after treating this TLR9 antagonist, it showed reduced level of untreated cells. So it suggests that this stimulated cells did release the mitochondrial DNA, and this mitochondrial DNA made a contribution to this self-induced netosis through the toll-like receptor 9 signaling pathway. After that, we developed a netosis propagation assay to mimic the neutrophil swarms at site of damage. So first we have NES plaque here. Those are stimulated cells. We stain them with host. That's why they look blue here in the image. And they will function as the early arriving neutrophils, which get stimulated by the pathogens. At the very bottom, we have untreated cells. Those are all naive cells. We used a piece of filter paper to immobilize the NES plaque on top, and we have naive cells at a very high cell density at the bottom. And we also added Setox screen here, which is a membrane impermeable DNA stain. So once the neutrophils at the bottom release NES, they have ruptured the membrane. The Setox screen could stain the cells and release NETs. Here are some results in figure A here within 20 minutes. Looking at this area, we didn't see much of NES release. We only have some dead cells have been stained with the Setox screen, those very bright green dots with sharp boundaries. But after 180 minutes, as shown in figure B here, for the regions close to the NES plaque, we saw lots of NETs released by stimulated cells. And but for the regions a little bit further away from NES plaque, we didn't see much of NETs released there. Those are just some dead cells have been stained in green. And we also have a control group here, as shown in figure C. Instead of using the NES plaque on the top, so we have the naive cells on top and naive cells at the bottom. And we didn't see much of NES release within 180 minutes. Then we developed an algorithm to analyze this spatial propagation of mitosis. We used the net score, which ranges from 0 to 1. We first remove the signals of the net plaque. So the center area here, we have 0 net score. And we found that the net score increased over time, especially in regions close to the net plaque. For example, region B1 here. But for regions a little bit further away from net plaque, we didn't see much of net score increase. For example, the region B2 here, the net score stayed at a very low level. If we take a look at the raw images from these two regions, the region B1 and B2, we found that in region B1, which is much closer to the net plaque, it released lots of nets over time. So the net score increased from 0.5 to 5.5 here. But for region B2, we didn't see much of net release. So the net score stayed at very low, around 0.7 here. And to assess the propagation pattern, we obtained the propagation profiles by averaging the net score over all radial directions at different time points. So as shown in figure A, the x-axis here shows the distance to the edge of net plaque, and the y-axis shows the averaged net score. After 20 minutes, it showed a radially decreasing net score distribution, moving away from the net plaque. And this propagation profile showed a strong fit in an inverse exponential model, as shown in figure B here. And we also found that after 140 minutes, the growth of net score decreased, which suggests a self-limit mechanism. In conclusion, we developed another one microwell method for the study of netosis. We proved the self-amplifying netosis by testing cells at different cell densities and by culturing stimulated cells and naive cells together inside nanowell unit. And also found that mitochondrial DNA released from stimulated cells made a contribution to this self-amplifying netosis through the toll-like receptor 9 signaling pathways. And in our propagation assay, we found that netosis could propagate from stimulated cells. So that's our net plaque there. And this propagation profile showed a strong fit in an inverse exponential model. So in the end, I want to thank everyone here, and I'm gonna pass it to Erik.
All right, thanks, Pan. Hi, everyone. My name is Erik, and I'm a PhD student in the Ma Lab. Today, I'm going to be presenting my recent research on assessing red blood cell deformability using deep learning. So imagine you have a little camera gun where you can take pictures of household objects, and it'll tell you if it's detected a cat. Now, what if I told you that a device like this exists? Would you be surprised? Visual images carry a wealth of information, and until recently, we were not able to utilize this information effectively. However, by utilizing new deep learning and computer vision techniques, we can train computer algorithms to predict properties of objects present in images, like cats or individual biological cells. And so this is where my research comes in. My PhD research utilizes deep learning and computer vision techniques to assess properties of single biological cells. And here I'll be discussing its use to assess the deformability of red blood cells. So why do we want to evaluate cells individually? Do these phenotypic differences matter? So population average cell measurements can miss information contained in cell heterogeneity. The behavior of cells in the tail of a distribution or in a small subpopulation may differ from the cells at the mean. Further, for bimodal cell behaviors, a population mean may poorly represent the majority of cells. Multiple measurements may be required to elucidate the different patterns of cell heterogeneity. Alternatively, single cell analysis can assess fine-grained cellular differences. Single cell analyses produce data that can represent cells in a high-dimensional feature space. And using data analysis tools, cell subpopulations can be partitioned in feature space. This information can be further assessed to test the significance of differences seen between groups of single cells. So one strategy to conduct single-cell analyses is by using biomedical microscopy images. Biomedical microscopy images of single cells contain lots of information, including morphological shape and structural characteristics that can be inaccessible to an expertly trained human eye. Using imaging to assess single cells can play an important role in understanding metabolism, understanding signal transduction, and conducting personalized medicine. However, conventional single-cell optical images are usually obtained and analyzed manually, vastly limiting their throughput. Through the recent technological developments in microfluidic microscopy and high-throughput imaging flow cytometry, vast amounts of cellular imaging data are readily available. Traditional microscopy imaging is usually manually analyzed. Improved image analysis techniques, including higher quality imaging and 3D image reconstruction, can be assessed using machine learning techniques such as support vector machines, clustering, and principal component analysis. Alternatively, high throughput imaging via microfluidics or imaging flow cytometry collects vast quantities of data, making these tools well suited for deep learning analysis. So deep learning techniques have unique advantages for analyzing big data and have fundamentally transformed how we acquire, process, analyze, and interpret imaging information. Deep learning tools are a subset of machine learning and are characterized by layered convolutional neural networks that excel at extracting features from image data. Recently, deep learning techniques have been adopted widely across scientific fields, indicating a paradigm shift away from traditional data analysis tools, especially in areas of image analysis and computer vision. So on the left here, we see an abstracted structural view of a neural network. On the top shows the connected layers in the network, and the middle shows how these layers are structured. The bottom image shows the kernel operator that passes over portions of the image to extract relevant image features. The GIF on the right shows an abstracted view of neural activations that produce the correct prediction of the number in the image. This is a classic example from the MNIST dataset, which is a dataset consisting of handwritten numbers, and this can be used as a benchmark to tune deep learning models. So why exactly do we want to assess red blood cell deformability? So the primary function of red blood cells is to enable respiration in tissues by providing oxygen and removing carbon dioxide via gas exchange in the lungs. During a typical 120-day lifespan of a red cell, they travel over 500 kilometers circulating through the body's arteries, veins, and microvasculature. During circulation, these cells repeatedly deform or change their shape to squeeze through the microvasculature, often squeezing through constrictions much narrower than the size of the unstressed cell. Therefore, the ability for the red blood cell to squeeze through small constrictions dictates its ability to facilitate gas exchange. Having a method to evaluate this deformability aids our ability to evaluate a variety of blood disorders such as malaria and sickle cell disease, or to evaluate the quality of a blood donation for transfusion. So we define red blood cell deformability as a cell's ability to squeeze through the microvasculature, and it's an indication of cellular age. As such, it's a biomarker for transfusion quality. More deformable red blood cells increase skin blood flow and hemoglobin levels, whereas sufficiently rigid or sufficiently aged cells get caught in the microvasculature and are targeted for phagocytic removal. Transfusion red blood cell deformability is dependent on both the donor's characteristics, such as their age, sex, diet, genetics, or activity level, and the donation storage time. The effect of storage duration on red blood cell deformability is also donor dependent, where some donors' blood stores better than others. Specifically, it is well known that the benefit from donated blood units vary, in part due to these donor-specific differences. We suspect that red blood cell deformability is a vital biophysical property that can be used as a predictor for transfusion lifetime. By predicting the longevity of a blood transfusion, blood banks could preferentially allocate blood units to patients based on their need. We hypothesize that chronic transfusion patients should receive longer-lasting blood to extend the time between needed transfusions, whereas acute transfusion patients, such as those suffering from trauma injuries, should receive shorter lasting blood as they just need a blood top-up and can normally produce healthy red cells. So here I'm going to describe my general experimental approach. So a red blood cell sample from a donor is processed and infused into a microfluidic device at 1% hematocrit. The cells are sorted by their deformability levels and a donor rigidity score is determined. After sorting, the cells are removed from the device and placed in imaging well plates. Using a Nikon microscope at 40x magnification, full well image scans are obtained. These full well image scans are passed through a custom computer vision segmentation algorithm and are augmented by rotating the images to produce balanced classes of different deformability levels. The training dataset is used to train a convolutional neural network, which learns relevant image features to detect the deformability of red blood cells using new and previously unseen images. Finally, the deep learning derived deformability score can be obtained and is compared to the microfluidic method. So a previous PhD student in our lab developed this microfluidic ratchet sorting device, and red blood cells are passed through the device to assess their deformability. Seen here in figure A, we have tapered funnel constrictions that allow for unidirectional filtration of red blood cells upward through the constrictions based on their tapered geometry and upward oscillatory flow. Downward oscillatory flow declogs cells that are too rigid to pass through the constrictions, and the constriction width for a row of tapers decreases along the flow path to enable deformability-based cell separation. Seen here in figure B is that the deformability sorting occurs in these matrix of tapered constrictions, and this produces a ratcheting effect. C depicts a 10x magnification micrograph of the active sorting region where cells are routed to different deformability outlets. And in D, we see a 4x magnification micrograph of the entire microfluidic sorting region. Red blood cells pictured here flow in an upward diagonal direction from the sample flow inlet on the left through the matrix of tapered constrictions and are primarily routed to outlets 3 and 4 on the right. So here I'll describe our microfluidic deformability-based sorting results. In A, we see red blood cell distributions after sorting for donors 2 and donor 4. Donor 2 is the most deformable, shown here in orange, and donor 4 is the most rigid, shown in green, of the 10 donors that we analyzed. Figure B displays the cumulative distribution of red blood cell deformability for these 2 donors, and the 50% crossover frequency is defined as the rigidity score for each donor. Finally, in image C here, we see the cumulative distribution plots for all 10 donors, indicating that there's inter-donor deformability differences. Now, figure 4 here describes a deep learning deformability classification. In A, we see normalized confusion matrices for donor 3 when the red blood cells from outlets 2 to 5 are considered as separate classes. We see substantial misclassification between outlets 2 and 3 and between outlets 4 and 5. In B, we have another normalized confusion matrix, but here we pooled donor red blood cells from outlets 2 and 3 together to create a deformable class, and we pooled cells from outlets 4 and 5 to create a rigid class. We see substantial increased accuracy after the data sets are pooled in this manner. In C, we see example images for outlets 2 to 5 for donor 6. We do not see any obvious visual differences in the red blood cells from these outlets, indicating that an expertly trained human eye couldn't be able to identify deformability differences manually. And in image D here, we see image classification results for training, validation, and testing accuracies for all the donors, and we see good consistency across the 10 donors with roughly 80% accuracy across the board. So oftentimes, machine learning models are described as black boxes. For instance, information is inputted and a bunch of complicated mathematical functions are applied to the data to learn these relevant features, and then the model spits out a prediction. All the while, the user does not see what these mathematical functions are actually doing. One worry of using machine learning tools is that the model could be learning on irrelevant confounders and not on relevant pieces of information. To confirm that the model is learning relevant features in the images, we applied saliency maps to the inputted images. These saliency maps displays which pixels in an image are relevant for the machine learning model to classify an image, where warmer colors indicate greater contribution of classification and cooler colors have lesser influence. It is clear from these saliency maps that the model learns based on cell morphological features in the images, suggesting our model is successfully capturing relevant information. So this deep learning method was applied to 10 donors with an even sex split and for a variety of blood types. The training data for all 10 donors converged, and testing accuracies ranged from 68% to 98%, with an aggregate average of 81 plus or minus 11%. The deformability curves and rigidity scores varied between the donors. As seen from these plots, The deep learning model successfully predicts a donor's rigidity score using data separate from its training. The aggregate average in rigidity score difference between the microfluidic and deep learning methods was 0.1 plus or minus 0.07. Finally, the deep learning rigidity score is plotted versus the microfluidic rigidity score. We find that there's high convergence, R equals 0.94, between the rigidity score evaluations using these two methods. So finally, the deep learning model was tested on cells that were not sorted by the microfluidic device. Applying the model in this manner is an example of transfer learning, which is applying the model in a different manner from its training conditions. Shown here are examples from donors 2, 3, and 4, clearly showing that the predicted rigidity score applied to this new data set provides similar deformability evaluation as a microfluidic device. So where do we go from here? We have a black 6 mouse study in the works where we look to track transfused cells using fluorescent labels. Blood draws are taken from the mouse at regular intervals post-transfusion, and the samples are put through the microfluidic device. We hypothesize that more rigid transfused cells are preferentially cleared, so over time, more fluorescently labeled deformable cells will remain, while more rigid label cells will be eliminated. Results from this mouse study will inform us on the feasibility of using deformability as a biomarker for transfusion lifetime. This will be further investigated in a clinical study with myelodysplastic syndrome chronic transfusion patients. Samples of transfusion blood will be taken and microfluidically sorted, and the rigidity score of the bags will be related to the time between subsequent transfusions for the recipient. This will inform us about the relationship between red blood cell deformability and transfusion lifetime for MDS patients. Finally, the deep learning deformability prediction method will be applied to donors while their blood is stored. We aim to evaluate how well the computer model can detect storage-related morphological changes. If the machine learning model can be made more robust, it'll greatly expand the access of red blood cell deformability evaluation to any lab or clinic with an adequate optical microscope. Thanks for listening. I'd like to thank all the people, research groups, and funding that made this work possible.
Hi, my name is Jayden. I'm a PhD student in MALAB. Today, the topic I'm going to talk about is insulin secretion and phenotype analysis of single beta cell using nanowells in microwells, as we talked about. So, understanding the heterogeneity of single pancreatic beta cell insulin secretion is important to discovering the fundamental mechanism of beta cell failure in type 1 diabetes. One of the approaches for measuring, observing the secretion in the single cell unit is confining the cell and bead in the nanowell, which can contain the nanoliter of reagent. So concept is pretty simple. So we are fabricating arrays of nanowell on the commercial 384 microwell plate. By doing so, we can utilize the automation and the observing process and the liquid handling process, which have been already made by commercial market. So we can have huge benefit of high throughput analysis of it. So after making the nanowell, we keep our cell, add our cell and detection measurement in the same nanowell, and then we we do the SS protocol to observe their secretion, and then we will get result. So the thing I'm trying to do is actually observing the insulin secretion of the beta cell in a single cell unit. So as we all know, the beta cell, when the beta cell is stimulated by the glucose, it secretes insulin, and then I as I mentioned, I put the measurement tool that can capture insulin on it, and then using the immunoassay to insulin, immunostaining to insulin, so we can make the bead capturing the insulin brighter. So depends on the amount of insulin that captured in the bead. The brightness of bead will be different. That's our hypothesis. So the matter is, how are we going to make the nanowell inside the microwell? And then how do we make the beads properly measure the amount of insulin? So let me just start first from the nanowell. So here is the process, microfabrication process for nanowells. Before we start, we should clean glass very thoroughly, and then we did hydroxylation treatment on the surface, and then actually the hydroxylation treatment is for the silent modification, which can bound the organic matter, which is polymer on, and then inorganic matter, which is glass at the bottom. So the silent modification gonna couple those two matter together, and then as we illuminate the laser, which is indicating here, laser patterning, the polymer is polymerized through the path of the laser, so that we can have the structure of the nanowell. And then after properly cleaning uncured pre-polymer, we can assemble those fabricated nanowell to the bottomless well plate, which is provided by commercially. So we can have the nanowell inside the microwell. So the major function of purpose of the nanowell is containing the cells and bead in the same position through the whole assay protocol. So here is the test that I did to find that the function of the nanowell is actually working or not. So just repeatedly aspirating, dispensing the reagent in it while the magnetic fields are applied. So here, I'm not sure you guys can see there's a magnetic bead, which is glow as lead, are deposit actually initially seated on the microwell, in the nanowell, and then while magnetic field is applied, they will hold them down, and we will see how many beads are actually left by picking those beads by image analysis. So those beads are autofluorescent and they glow as lead, so we can pick it. So we can actually count their number before and after. So there are some imaging errors in imaging process, as we all see. Like if I have contaminants like this, it will create some errors. Like the highest lost rate is like about 5% of beads are lost. So I just with my bare eyes, I check that, and then I confirm all beads are probably securely sit in the same position. Probably you can compare with your bare eyes too here. So as I mentioned, another matter is beads. So to see beads are working properly as the amount of the insulin captured, so we did doping test. So it's basically just doping insulin to the beads in different concentration, and then through the flow cytometer, we have checked the beads are properly worked, and then when they are distributed in the nanowell, and then we have, if we average them, I mean, we average the value of the brightness per nanowell unit, we will have those kind of clear standard curve. We confirm it. That's we confirmed. So those one dot indicating average value of 1 nanowell. So in this case, just picking all the beads together and then flat it. In this case, it's averaged by the nanowell. So we got two nanowell. We got proper nanowell, and then we got proper beads. So now we can do the glucose-stimulated insulin secretion assay protocol. And then here's the protocol that I did with the stem cell-derived insulin secreting cell. So, yep, cells are dissociated and then resting in the colorless buffer for 30 minutes, and then exchange the media to PBS, and then just filtering them through the sieve that we can have single cells. And then the single cells are being dispensed in the microwell, so they will be distributed each nanowells at the bottom of the microwell, and then the cells are stimulated, and then these are immunostained. So here is the image processing that I used. So basically, when I captured the images, I manually picked the nanowell with a single cell with multiple beads. So comparing two pictures before assay, after assay, we check the all beads and all cells are in the same position. So after verifying that, we measure the brightness of the beads per nanowell unit. So that's how we get result. So this is one example of the cell secreting insulin. So the picture on the left side is before, and then right side is after the assay. You may see on the right picture, some glowing beads as orange. So when the beads are when the beads captures the insulin, and then when they get immunostained, they will glow as orange. So basically, it is showing the green cell is secreting insulin. So the nanowell is actually confining those flow, and then we will see like Gaussian graph here. So I have this is result I have recently got, and then so all those flats are also, as I mentioned, one dot is indicating 1 nanowell. So one dot is just average value per nanowell. So we can see clear here. You can see clear difference, high and low glucose level, and then the amount of insulin secreted by we can see difference here and here. And then the dioxide is used for prevent the cells insulin secretion. And so here, those 3 data set are technical replicate, and then even there are some confusing result in 0 millimole glucose concentration. But the thing is, we can see the difference is low glucose level and high glucose level. here and here. So K cell is used for positive control. So this research proposed the nanowell-based platform to measure the heterogeneous of single beta cell insulin secretion, and then we fabricated high aspect ratio of nanowell that can contain cells embedded in the same position through the whole assay protocol. And so high density of nanowells capable of profiling multiple single cells simultaneously, and then they are highly compatible with the standard high throughput infrastructure for microscope and then liquid handling. So yeah, these properties provide significant advantage for microscope-based analysis for single cell heterogeneous phenotypes. This is the end of my presentation.
So yes, we'll take some questions. So
this work. Oh, great presentation. I have a question for Erik for his machine learning. What is your baseline that you're checking your machine learning towards? Like, are you checking red cell functions? I know you're doing the microfluidic device, but how do you make sure that the cells, let's say, the cells that are functioning, and your machine learning is capturing that completely? Like, what is your baseline that you're checking against?
Sure. Thanks for your question. We don't do a general red cell functioning test, but we get red blood cells from self-professed healthy donors. And what we do is we process those samples, remove the plasma and leukocytes and platelets, and then infuse them to the microfluidic device. And that provides our true label. You know, did this cell go to outlet 2, which is very deformable, or outlet 5, a little bit more rigid. And those are defined as like the true classes for each cell. And then those cells are extracted out of the microfluidic device, imaged, and those true labels are what the machine learning model is testing against. So machine learning model gets a lot of cells and it predicts, you know, is this cell image that I have, like, is this an outlet 4, for instance? And if that's a wrong prediction, it'll be told the correct, you know, it's actually an outlet 3. And then the model will update its parameters based on that feedback and slowly get towards a convergent solution where it can, you know, with high accuracy predict the deformability level of each cell. And so we hypothesize that the different deformability levels are kind of proxies for cell function. The more deformable they are, the better they are at navigating through the microvasculature. The more rigid they are, maybe the older they are, and the sooner they'll be eliminated. So that's kind of our proxy for quality. And we're doing a few other studies to evaluate a little bit more of those mechanisms. Thanks.
I have a question for you, Erik. I'm curious to know if when you did your storage studies, were you storing all those units to the same date, at the same length of time?
Yeah, that's a good question. The answer is no. I have it all here. So these are the donor characteristics. So we had an even sex split and a bunch of different blood types. Most of our samples were generally fresh, stored for 0 or 1 day, but some were longer at day 12, day 9, and day 23, and in a variety of conditions, blood bag or in accelerated storage in a tube. So we do have a wide variety of different storage conditions here. And some of the future work we're doing is testing this model on defined intervals, just like Amel's previous study, testing it at day 0, week 1, week 2, seeing if the model can predict how long these cells have been stored for, kind of looking at how much degradation is occurring there. And instead of doing live cell imaging, extracting the cells directly from the microfluidic device, we're doing blood smears, which give us much higher quality images and is closer to what is done in clinical practice. So hoping, blood banks could take a blood smear of a donor and get a quick measurement of what's the quality of that blood, how deformable is it, how long has it been stored for, things like that. Does that answer your question?
Yeah, it's probably better done by the blood operator looking at a blood system to look at these donor factors, but it is something that could get done in a hospital blood bank. The other question that I have for you is, it's just a fact that the DEHP, which goes into the red cell membrane, stabilizes it to a certain extent, which allows us 42-day storage in North America. This compound has been banned in Europe now, and so all of the blood bag operators are getting rid of it, and so they're bringing in different plasticizers. And I think that your assay would be really interesting to look at with these different blood bags.
Yeah, that's a really interesting point. We'll have to think about that and investigate a little further. Amel has done some research looking at the degradation of red blood cell quality in blood bags versus in tubes, and find that the plasticizers in the blood bags retain the deformability to a far greater extent than in tubes. But it'd be interesting to see the different degradation, I guess, profiles of different plasticizers, and maybe some donors are really good with some plasticizers and not with others.
Yeah, that's what we think we're going to find.
Interesting. Cool.
So it's one of my further step is so now we are stage of just looking at phenotyping. They are secreting a lot of insulin or less amount of insulin, but eventually our goal is extracting those cells from the non-well and then and then doing other genomic analysis. So what makes them different in this insulin secretion, and then that information would give more benefit of food.
I can extend on that a little bit. So right now, the central derived cells do not produce nearly the same amount of insulin as the primary beta cells. And so our sort of larger hypothesis is that perhaps within those cells, some of those are better producers than others. And the question would be, how do you get more of the better producers in the beta cell?
You guys did a great job, all of you, including Dr. Ma. It's wonderful to be able to see the development of new technologies. It seems only appropriate that we also have a question for our NETosis expert. It's wonderful that you captured a field in a single plane for your fluorescence. But I'm wondering if you could also benefit by also getting some data in also the z-axis, so that maybe because of course nets are not two-dimensional, they're three-dimensional. So I'm wondering if you might increase your resolution, increase your sensitivity, and also increase your accuracy then, because you're gonna get a better picture then. That's a pun, by the way.
Thanks. It's really hard to find the nest for sure, and we do need lots of higher concentration of stimuli to get lots of nests. And for nest plaque, in our NETosis propagation assay, just to mimic as the earlier arriving neutrophils, there are 3D structures, but it would be hard for us just to get a whole picture of the 3D structure, so that we don't know how they contact with the bottom layer of the cells. But it would be really great that we could get 3D structures of the nets, so we can better study whether it's because of net cell contact or because of just some diffusible chemicals received from the naive cells so they could get activated.
Thank you.
Thank you.
Hi, thank you. Thank you for your presentation. Also another quick question for Erik. You showed a slide showing two confusion matrices, and you mentioned that grouping the outlets together produced a more distinguished segmentation. And I was wondering, what's the scientific basis for grouping the outlets?
Yeah, thanks for your question. So you're talking about the plots on the left here. So the basis for this is that we evaluated 10 donors, and not all 10 donors had cells sorted to outlets 2 to 5, but all donors had cells at least in outlets 3 and 4. So we were able to, we found that, yeah, by grouping outlets 2 and 3 together into a deformable class and 4 and 5 together into a rigid class, it increased the accuracy. And that could just be due to, now it's a binary classification, right? You're now classifying, the baseline, a random guess is 50%. So getting 80% accuracy, that's only 30% above baseline, right? If we had the other 4-class classification, baseline's 25%. So, 55% would be the same increase in accuracy. But we found that for purposes of comparing different donors to each other, when not all the donors had cells sorted to all these outlets, pooling them in this manner made it so that we had consistent quantification analyses at the very end for what is a rigidity score for each donor. We could apply the same method to all donors, and hopefully that'll get us to a point where we can evaluate red blood cell deformability for any new donor, because this is now a standardized implementation that should be applicable to every donor. And we didn't see a decrease in, or a substantial decrease in accuracy of those final rigidity score results. We still found, a difference of 0.1 aggregate average difference between the microfluidic and deep learning derived rigidity score. So we didn't lose a lot of accuracy in this approach. Does that answer your question?
Sure. You mentioned that all the donors had at least had been in either outlet 3 or 4, but does that explain why you grouped 2 or 3 and not, for example, 3 with 5 and 2 with 4? Is that?
Oh yeah. So outlets. So the difference between a cell being sorted to outlet 2 and being sorted to outlet 3 is a difference of a gap. So there's small little tapers that the red cells are squeezed through. And I don't know it exactly off the top of my head, but I think the gap between at outlet 2 is around 2 micrometers, and for outlet 3 is 2.25 micrometers. So the difference is very minute. And then when you have, so the difference for each of the class is an increase or decrease of 0.25 micrometers. So, we want to group the deformable ones together. Outlet 2 and 3 are more similar than 2 and 5 because the gap there is 0.75 micrometers.
Okay, thank you.
Yeah. All right, I'll take that one for time.